Hi everyone, today we're going to be talking about echinoderms, as we can see in this picture by these beautiful crinoid fossils. But if you don't know what a crinoid is yet, don't worry, by the end of this video you definitely know what crinoids are. So let's get into some background on the echinoderm phylum of invertebrate animals. Echinoderms are animals that include things like starfish, as we can see in the middle two pictures here on the left. We have a living one, a fossil one. They also include things like crinoids, as we saw in the previous slide, which are also shown at the bottom here. We got a living one and a fossilized one. They also include things like echinoids, or heart and sea urchins, and sand dollars, as shown up here in the top. We have a living urchin here in the middle with all its crazy spines. We have a fossilized one on the right, and we have a sand dollar to the left. And they also include things like blastoids, which aren't pictured here, but we'll talk about what blastoids are on a later slide, and pretty much any other invertebrates with 5 sides. With this, that brings us to our first common characteristic we see among echinoderms, and that is that they have pentagonal or radial symmetry, as we can see pretty easily with the starfish here. But also, if you were to take apart this crinoid down here, or echinoid, or sand dollar, you would see they all have 5-sided or radial symmetry, and that is a common characteristic among echinoderms. Also, in terms of ecology, they can feed in a variety of ways. They are very diverse. One of these ways is attached echinoderms, such as crinoids and blastoids, that feed as filter feeders, or sand dollars, heart urchins, and sea cucumbers that feed as detritus feeders, and sea urchins that graze algae, and sea stars that act as active predators that feed on clams, barnacles, or corals, and brittle stars that can either deposit feed, scavenge, suspension feed, or feed by predation, and lastly, bracket stars that can catch large prey. So there are a variety of ways that echinoderms can feed, and obviously a variety of diverse organisms that are included within this phylum. However, something that echinoderms don't include are planktonic forms. They also don't really have many nectonic or swimming forms either. However, certain kinds like stalkless crinoids, as shown in this GIF here, can swim for very short distances. And as we see, it is really beautiful, and if you want to see these guys swimming like this, you can look up videos, just type in something like feather star swimming, because a lot of times, because they're so beautiful when they swim, they're called feather stars. Next, I want to jump right into echinoderm morphology. Some additional common features among echinoderms, rather than just their symmetry, include things like their skeleton or test, which is made up of calcite plates or ossicles, and many, but not all, have spines, and that's actually what they're named for. Echinoderm means spiny skin. And if we move on to some of their more specific morphological and anatomical characteristics, we see here that they have a water vascular system, which is a network of tubes and canals controlled by hydraulic pressure. And so we can see this in this picture to the right with this starfish with all these tubes in it. That's because it has this water vascular system. And they have a madreporite, which is a plate on top of the animal that regulates the fluid pressure. And they also have a stone canal that connects this madreporite to the main pipeline of the animal. As we see this ring here in the center, that's the ring canal that kind of connects all these radiating tubes of the water vascular system to each other and to connect the whole body of the animal together in its entire system. And another characteristic is that they have these tube feet, which are connected to the end of each radiating canal. So each arm of this starfish has a radiating canal, and shown on this arm, but they're on every arm, are the tube feet, which are shown in close-up here at the bottom. And these tube feet can be controlled by this ampulla here, and the ampulla is this bulbous part of the tube foot that has a muscle that, when squeezed or tightened, will protrude the tube foot, and the starfish or echinoderm can then move around, and in the case of starfish, actively prey on other animals. And so these things encompass the general morphology and anatomy of echinoderms, but we will talk in more specific terms near the end of this lecture about crinoid and echinoid morphology specifically, because they have a lot of little features that really help with identification. And so if you want to understand more about crinoid and echinoid morphology, stick around and we'll talk about that in more detail later in the lecture. But first, I want to talk a little bit about how echinoderms are similar to us. You might think this is crazy because we just saw a list of organisms that are nothing like us, but actually echinoderms are our closest invertebrate relatives. Because echinoderms are so closely related to chordates relative to all of the other invertebrate phylum groups, echinoderms and chordates are lumped into a group called deuterostomes, and this is because they share similar larvae larva that differs greatly from the larva shared by other invertebrate groups. And this is why echinoderms are more similar to us than any other invertebrate phylum. So now we can move on to classification of echinoderms. Here I want to go over 12 classes of echinoderms. Yes, 12. You don't have to remember them all. We'll focus on the crinoids, echinoids, and blastoids for the rest of this lecture. But I do want to stress that some of these classes may be named slightly different or lumped in with each other slightly different than you might see in your book, because every book differs. If you want to see the book that I'm using to make this lecture, it is always linked down in my description, so you can check that out if you'd like. But for these classes, we have the crinoids or crinoidea. We also have eocrinoids, paracrinoids, blastoids, like I mentioned previously, rhombifera, and diploporida. And these are a couple that on my book are lumped in with each other, but obviously are different things. So in your book, they might not be lumped together, or maybe they're lumped together and they're named something different, but this is what they generally look like. Also, we have Stylophora ophiuroidea, I hope I'm saying that right, Astroidea, Holothoroidea, which as you can see include sea cucumbers, Echinoids, as we also previously mentioned, Eoastroidea, and Helicoplacoidea. But like I mentioned, we're going to be focusing on crinoids, blastoids, and echinoids. So first I want to briefly go over blastoids, which went from the mid-Ordovician to the late Permian period. So yes, they are extinct, but they are really important in the fossil record because they can be really good index fossils, and they have a really distinctive morphology. So let's go over what their morphology is. Similar to crinoids, as mentioned earlier, they can have stalks and therefore be attached to the bottom sediment in the ocean, and they also have a symmetrical head, or what's called a theca. However, unlike crinoids, as we'll see later, they have unbranched arms, which are called brachioles, and their arms are very rarely preserved. So you don't actually see those in, for example, this picture to the bottom or right, or even in the morphological figure, because their arms are so rarely preserved that they just don't feel like they should show it, because it's hardly ever a thing that we get to use to identify these things. However, even without their arms, they're pretty distinctive, and so some of these distinguishing features are circled here and listed over here to the right. These include 5 ambulacral areas, and these are shown here on this image. We have these things that look almost leaf-like on each of the 5 sides of this organism, and those are the ambulacral areas. And in life, these were covered by plates that closed the pores, because as we can see down here, they did have pores in their body called inlet pores. And behind each of these ambulacral areas, they had folded, pleated hydrospires shown at the bottom here. This image shows the theoretical during life hydrospire that would have been there behind those areas, and that structure was used for respiration. And lastly, at the top of these animals, they had spiracles, which are these holes up here. Basically, these were the water outlets. And we can see circled in orange here, these little tiny holes along the sides of the ambulacral areas were the brachial sockets, where the arms would have attached. So what about crinoids? These beautiful, beautiful swimming or crawling organisms that we see in these images. So to get into crinoid morphology, we have here a figure showing different crinoid features, and we're going to go over these one by one now. So the first thing we list here is a head or calyx, which is shown in the image all the way to the right here. This calyx includes this whole fat part right here, and this is basically what we call the head of the organism. And then we see the mouth, which is pointing over here. It's hard to see this line, so it's within the center of the top of the calyx, and this is actually what we call the oral end of the animal, as opposed to the stalk end, which is the aboral end of the animal, obviously because it's on the other side of the mouth, whereas the mouth is on the side of the mouth. And on the oral side, we have attached to the crinoid's head its arms, which, like blastoids, unfortunately, are less commonly preserved than the rest of the animal. Typically, what you'll see in the rock record is actually just the stalk, and it's not even the full stalk. Most of the time, you find crinoid stalks that are broken up into a bunch of little pieces in the rock record, and you can hardly even identify the species, let alone major group of crinoid it is, because all you have are some plates of the stalk, and you don't even have any of the head or the arms. But when you get the head and the arms, that's why these things can become very valuable, because That's rare. Next, we have pinnules, which are labeled up here, and these pinnules are the branches, if you will, of the arms, which help to increase the surface area of the crinoid arms and therefore its fluttering ability. Or as we saw previously, the swimming crinoids, they wouldn't be able to do that if they just had an unbranched arm that didn't have a wide surface area in order for it to help it swim. So then we move on to the cirri, which is shown on the stalks. These little extrusions from the stalk are called cirri or singular cirrus. They're also shown over here in plural. And moving to the plates of the crinoid head or calyx, they have a series of really distinctive plates. And this is going to be the most important thing for crinoid identification. Here we can see that these radial plates are labeled at the top or the oral end of the crinoid's calyx. And down toward the bottom, we have two different crinoid calyces or heads shown down here in C and D and E and F respectively. C and D is indicating what's called a monocyclic crinoid. And this means that this crinoid has radial plates and then basal plates, and that's it, and it doesn't have anything below or aboral to the basal plates. The basal plates are the most basal plates on a monocyclic crinoid. However, there is also something called dicyclic shown in E and F, and these have what's called infrabasal plates below the basal plates, and so these guys are then termed dicyclic, and we'll see later some more different plate patterns of crinoids because they can unfortunately get kind of complex. So we'll go over those later, but in simple terms, the important series of plates include the radials, the basals, and if applicable, the infrabasals. Now, sometimes crinoids can have what's called fixed brachials or plates above the radials, which aren't shown in these images, but we'll show those in a couple slides. And they also sometimes can have interbrachials or plates between the brachials and the radials. And we'll also show those on a later slide. Lastly, for this image, we have labeled here the tegmen, which is basically like the roof of the oral side of the calyx of the crinoid. And then we also have columnals, which are each little segment of the crinoid stalk, and these are often what's broken apart in the rock record because they have these columnals and they can break along those edges, and so they often do. And then lastly, we have the holdfast or the root of the crinoid stalk, where it can attach to the bottom sediment. Now, like we saw previously, some crinoids can be stalkless and therefore wouldn't have a root and wouldn't attach to the sediment surface and would actually be those short distance swimmers like we saw in those GIFs. Now, before we get into the more specific details about the plates of the crinoids, I need to mention the four main types of crinoids, and these include the oldest of the four types or most primitive, and these are the inadunates, which have rigid calical plates or just plates on their calyx, and they have no fixed brachials or interbrachials. Those kinds of plates that are above the radial plates evolved later when crinoids started becoming more complex. Then we have the flexibles, which usually do have brachials above their radials, and then they also have both basals and infrabasals, meaning most of the flexibles are dicyclic. And they're named because their multiple calical plates are loosely united, and they have a flexible tegmen with an exposed food groove and mouth. And last, for the flexibles, they have arms with no pinnules. Next, we have the camerates. The camerates have a rigid calyx, a food groove that's covered by the tegmen, and multiple fixed brachials and interbrachials. Lastly, we have the articulates, the only class that survived past the Permian extinction and has continued to not go extinct until today. And this is the type of crinoid that we see in the modern ocean. The articulates have very flexible, articulated arms, which is what gives them their name, and they also typically have 5 infrabasal plates. So here's a figure that I just want to go through one more time to talk a little bit more in detail about these crinoid plates. Like we already mentioned, we have monocyclic types of crinoids, which have basal plates in this light red color and radial plates in this yellow color. And then we have dicyclic, in which they have the basal and radial plates, but below the basal plates they also have infrabasals. However, if we move over to the very left image on this figure, we see some more complex complex plates jumping into the game. What we have here are our normal radials indicated by R, and these are shown on the crinoid here. And below that, that's kind of hard to see because of the stalk, there are basals and no infrabasals. So in this case, this is a monocyclic calyx. However, above the radials, there are a series of plates that are also labeled. So these are, like we talked about, fixed brachials and interbrachials. The fixed brachials are the plates indicated by the BRs. This crinoid in particular has 3 sets of brachial plates labeled BR1, BR2, and BR3. And the plates in between the brachials and radials are the interbrachials. So we can see one of these shown here on the image at the top left. So now that we have that understood, I want to try and label with you guys some major types of crinoids just based on their preserved calyx. And so we're going to look at the plate structures here for both monocyclic versus dicyclic, as well as the 4 main types of crinoids we went over, because this is likely something you might have to do for a class. So these 5 crinoids are labeled here, and we can tell that this first one in figure A is monocyclic because it has basals and no infrabasals. We can tell that the next one down is dicyclic because it has infrabasals and basals. Then we have monocyclic again with just basals, no infra. Then we have dicyclic again with infrabasals and basals. Then we have monocyclic in the last one with just basals. Now to go over the 4 main types of crinoids, first we have inadunates, which just have radials and don't typically have any brachials or interbrachials. And so we can see that this one follows mostly that rule. It has some brachials, but the radials almost go to the top, and it has very few brachials compared to all the additional brachials and interbrachials that the rest of these crinoids shown here have. Then the next one we can tell is flexible because it has 3 infrabasals, which is pretty typical for flexible crinoids. Then the next one we can tell is camerate because characteristically it has multiple brachials and interbrachials. As we can see in this crinoid, it's got so many plates above the radial plates, and so that's pretty typical for camerates. Then we have the dicyclic camerate shown down here. It also has a lot of interbrachials and brachials. And lastly, we have the articulate crinoid shown at the bottom, which has typically 5 basal or infrabasal plates as shown here. However, articulates are more easily identified with more certainty if you have the arms preserved, because like we mentioned, they have highly articulated arms with extensive pinnules. Now to go over a little bit about crinoid ecology before we move to echinoids. Crinoids are pretty sparse today, but used to be super abundant in the ancient ocean. Modern stalked crinoids live in typically deep water, around 100 to 5,000 meters in depth, and modern stalkless crinoids live in a little bit shallower water and are more mobile because they can swim a little bit, and for that reason they can avoid predation, which is why they're able to live in shallower waters and not be eaten up, or at least not as often as the stalked crinoids would. However, Paleozoic crinoids happily lived in shallow waters, even attached to the surface with their stalks. This was because they didn't need to have that fast of an escape mechanism because there weren't as many predators to the crinoids back then as there are now. And for this reason, we can see that crinoids made up something during the Paleozoic called crinoid gardens, as we can see in this image, and that would have been in the shelf lagoonal environment of oceanic margins, and that would have been really cool to see, honestly. I wish we could have been there, but we can sort of go back in time if we pick up any rocks from the Mississippian. If you go out and look at Mississippian aged sedimentary rocks, you will surely find some crinoid stalks within those rocks. So I encourage you all to go look for some. Crinoids are super abundant during this time. But what about echinoids? Echinoids, as we can see here by these gifs, are really cool organisms. So let's talk a little bit about their morphology and how we can identify their fossils. As shown here, we have some common features of echinoids listed here and also identified here on this image. Echinoids, like many echinoderms, had tube feet, but in addition to tube feet, they often had long stalks terminated with pincer-like claws called pedicellaria. However, these are not identified on this figure. What is identified on this figure is Aristotle's lantern, which is this circled figure shown here labeled E on this image, and we can see Aristotle's lantern in the organism in figure B, and this apparatus helped them, or continues to help them, graze on algae. It is often thought of as a dental apparatus. You either jaws or teeth or kind of both, and it's even sometimes venomous. So be careful when coming in close contact with echinoids and their Aristotle's lantern. But moving on to their madreporite, we already saw this labeled on the first sea star we looked at, in which it helped to control fluid pressure because they have a water vascular system, and in echinoids it has a similar function. Next we have the paraproct, which is where the anus is located. Then we have the ambulacrum, which is shown up here at the top of the image, along with ambulacral plates and pores, and these are similar to what we saw in the blastoids. These are 5 segments shown here by the 5-sided symmetry of the animal in figure C, and these have both plates that cover them and little tiny pores on these plates that would have helped with fluid intake. Next we have the peristrum, which is on the opposite side of the paraproct and anus, as we can see at the bottom here where the Aristotle's lantern is, as well as the mouth. Next we have the genital plates shown here surrounding the paraproct of the animal, and then we have the ocular plates, which are tiny little plates at the end of each ambulacral area. And last we have the mouth, as we mentioned, which is where the Aristotle's lantern is. Next I want to go over some echinoid ecology. As we can see in this figure, echinoids can range from very flat to not so flat. Flatter echinoids are typically more advanced as they evolved later and have more functions, such as burrowing, which fatter and more primitive echinoids actually don't do. And these flatter echinoids, such as sand dollars as an example, are infaunal because they burrow into the sediment, and they evolved later in the Paleocene, whereas fatter echinoids were and often still are epifaunal in that they don't burrow into the sediment. They live on the sediment floor rather than in it, and they dominated before the Tertiary, or before the Cenozoic era in general. And these groups of echinoids, fat versus flat, equate to what we call regular and irregular echinoids. And the regular ones are the fatter ones, and the irregular ones are flatter, even though that's not irregular, it's just that it evolved later. And they also differentiate themselves based on environment. And this is where we get into ecology. So as we can see, these regular and irregular echinoids separate themselves based on environment. Basically, regular echinoids live in rock crevices in the intertidal and subtidal zones, as shown up here further inshore, and the irregular or flatter echinoids, like sand dollars, live in shallow to deep burrows, as shown here in these burrowing images at the bottom, and they live in subtidal to lagoonal areas, so a little bit deeper in the lagoonal subtidal area, as we can see in this burrowed echinoid here. And now finally we have the evolution slide. So first I'll go over the general Echinoderm classes and their evolution, then we'll talk about echinoids specifically, and then crinoids specifically. So first we have some experimental groups of Echinoderms evolve in the early to mid Cambrian and then go extinct because they were, as we say, experimental groups, and therefore they evolve into more complex and generally more robust groups. These include the helicoplacoids, stenocystoids, and homostellian echinoderms. Next, we have some Ordovician groups evolve, and by some, I mean quite a bit. These include classes Eucrinoidea, Paracrinoidea, cystoids, which are often clumped in with the diploporans, which also evolved during this time, the rhombiferans, the astroidea, ophiuroids, and possibly even holothurians, which are things like sea cucumbers, but it's hard to know because these are pretty soft-bodied animals that have a lack of hard parts and therefore are hard to find preserved in the rock record. Also, before we move on, it's important to note that the Ordovician was the peak of echinoderm diversity. Next, we have the cystoids and carpoids, which can be grouped in with the stylophores, go extinct during the late Devonian mass extinctions. Then we have the Permian mass extinction, causing a lot of extinctions, but among the most important was the extinction of the blastoids. Now moving on to the echinoid specific evolution, we have echinoids evolve in the mid to late Ordovician, and then we had the Permo-Triassic extinction that left only one surviving genus of echinoids. However, this genus was able to pick back up in the Mesozoic era and radiate once again, and unfortunately, even after all that diversification, it was hit hard again in the Cretaceous extinction. However, they came back to diversify in the Cenozoic, as we see with with flatter echinoids or irregular echinoids evolving in the Cenozoic, namely the sand dollars, which evolved in the Paleocene. And unfortunately, this arrow leads to a blank white space on this figure because I don't have the Cenozoic shown here, but you get the picture. After the Mesozoic, during the Paleocene, the sand dollars evolved, and then we had echinoid peak diversity, which is now. Next, we have crinoid evolution. Now, crinoid evolution starts with some uncertainty. First, we have the genus Echinoderms evolve in the Cambrian. However, it's unknown currently whether this Echinoderms is actually among the crinoids. It was first named because it was thought to be part of the crinoids, but now it's been grouped into the corals or cnidarians. But more certainly, we do have the inadunate crinoids evolve in likely the early Ordovician, which then led to the flexible and camerate crinoids evolving in the mid to late Ordovician, and then crinoid peak diversity, like I mentioned earlier, in the Mississippian, where we would have had those beautiful crinoid gardens. And then the next event, not shown here just because of lack of space, was actually these three major groups of crinoids, the inadunates, flexibles, and camerates, going extinct at the end of Permian extinctions. However, they came back with the articulates, which evolved in the early Triassic and then diversified after that. However, they never reached the same level of diversification and abundance as we saw in the Paleozoic crinoids, and this was likely because of higher predators evolving in the Mesozoic era and therefore not allowing crinoids to reach that same level of abundance in shallower areas. And so then crinoids later in the Mesozoic and Cenozoic and to now have survived, but these stalked forms have been pushed down to deeper waters, and the stalkless forms still can live in shallow waters but are less abundant. So that is it for today, guys. I have shown on this slide the upcoming videos for this playlist. We have the last two videos for the invertebrate paleontology groups, and these include the forams coming up next, and then the rest of the major microfossil groups, the diatoms, radiolarians, and coccolithophores, in the video following that one. And lastly, if you are still watching this video, comment down below your favorite echinoderm. Mine personally are these spiny regular echinoids. I think they are so cool with their little spines and their 2 feet moving around. That is just so cool to me. So anyway, comment that, leave your video suggestions, go watch these other videos. If you're interested in any of the upcoming content, please subscribe. And with that, thanks again for watching, and I can't wait to see you guys next time. Bye. Almost did that without recording. I was on a roll until I got to Ophiobroida and Helicoplac. It's not hard, I just get messed up. I thought the Tegman was the head. I'm like not remembering this right. And get the book. Yeah, those of you out there who think this is my brain, no, it's this. Penguins, where are you? Oh, oh darn it. Inadunates? Alright, well nobody wants to pronounce it for me, so why did I do that? I think my computer's about to burn up because I've been recording so freaking long. Whew, that was a long one.